All right, welcome to the podcast that covers Objective 3 for Day 27. In this podcast, we're going to talk about this equation called the Hardy Weinberg equation and the Hardy Weinberg principle, and how we can use this equation to test whether or not a population is evolving. And I know when I was a student, I remember every time I was in a biology class and they started talking about equations, I started to get nervous. Well, I don't want you to get nervous. I'm going to walk you through how we use these equations, and hopefully you'll see they're not as complicated. And we'll also do an activity in class too. Let's first review the terms population, gene pool, and allele. So remember, if we had this area here, and we had these species living together, same species interacting together within the same environment, and they can breed with each other and pass on their traits. So that's a population. We know that within this population, any one of these individuals is going to have a genotype. But we're just going to be simple and use the letter A. And we could have an AA, we could have an A1, and a small a. We could have a, big A, little a, A3, A4, A6, A7, A8, A9. Each one of these organisms will have two alleles for each gene. Those different alleles may ultimately come along and create different phenotypes. So maybe this one's going to be red. Maybe this one will be green. Maybe this one will be black. Maybe this one will be blue. Maybe this one won't have any color. Maybe this one will be polka dot. Right? So each of these allele pairs will generate a different phenotype. All of these alleles together, A, A1, little a, A3, A4, A6, all of them together, they constitute the gene pool. Population, gene pool, and allele are really important to understand them as we move forward, thinking about whether or not a population is evolving. So let's start off with a problem similar to the one that you see in your book. And actually it's the problem in your study guide, your note-taking guide. So a flower is determined by two alleles. So let's start by calculating allele frequencies. And let's work the problem that's on your note-taking guide. Your note-taking guide says that we have two alleles, CR and CW. And that these two are important in determining flower color. In this problem, we're told that we have 600 red flowers. I take that back. I'm sorry. We're told we have 640 red flowers, and that we have 320 pink flowers, and then 40 white flowers. So we have a total of 1,000 flowers in this population. Let's go ahead and write their genotypes over here. That might prove helpful later. A red flower is CR, CR, and a pink flower is CR, CW because of incomplete dominance, and the white flowers are CW and CW. If we have 1,000 flowers and we know each flower has to have two alleles, we know that we have 2,000 alleles. So let's just write that here. Okay, so now let's solve the problem. How many CR alleles, or what's the percent? CR percent. Well, first we have to do is look at our population here and determine how many CR alleles we have. And we can look at our red flowers. For every red flower, we have two CR alleles. If we have 640 red flowers, then that means we have 640 plus 640 to account for the red flowers. Now let's look at the pink flowers. It's a heterozygote, CR, CW. So for 320 pink flowers, we know we have 320 CR alleles. So we'll go plus 320. And then we need to divide that by our total number of alleles, 2,000. So 640 plus 640 is 1280. 1280 plus 320 is 1,600. Divided by 2,000. And that's 80%. So we know our population has 80% CR alleles. So let's quickly look at CW percent. What is it? Well, we don't have any white alleles in the red flowers. We know in the pink flowers, they're heterozygotes, so we know we have 320. And the white flowers, for every white flower, we have to have 2 CW alleles. So that's going to be 40 plus 40. So 320 plus 40 plus 40. And we're also going to divide that by 2,000. So this equals to 400 over 2,000. Or 20%. So the problem's answer is 80% CR alleles in the population and 20% CW alleles. Let's move on now and start talking about Hardy Weinberg. These are two separate individuals, not even really in the same field, that independently, they never knew each other, at least before they made the equations. I'm not real sure if they ever met, but they came up with something that we now know called the Hardy Weinberg principle. The Hardy Weinberg principle states that as long as there is no evolutionary force, and we'll talk about that in a moment, what we mean by that, then allele and genotype frequencies remain the same from generation to generation. So let's go and write that down too. From generation, we will often say that a population is in Hardy Weinberg equilibrium. So a population that is in Hardy Weinberg equilibrium is a population that is not evolving in relation to the genotype. When we do a Hardy Weinberg equation and apply it and ask whether or not it's in equilibrium, we only look at one genotype at a time, one gene pair at a time, at least at the level we're going to talk about today. So if a population is in Hardy Weinberg equilibrium, then that population is not evolving. So let's wipe this off and let's write the Hardy Weinberg, I'm just going to write H-W, Hardy Weinberg equation. And it's P squared plus 2PQ plus Q squared. And it equals 1. P squared and Q, well, P and Q refer to the alleles. I usually see P squared referring to the homozygous dominant. So maybe, let's just give an example here, like AA. The members of the population, which are homozygous dominant, big A, big A. And then Q squared would be little a, little a. And then the heterozygotes are PQ. Would be like big A, little a. And so we put a 2 here. And let's ask why we put a 2 there. That's because in any given population, if you have 2 alleles, you know there is a certain probability that'll be big A, say from the sperm, and a certain probability it'll be little a. It's not going to be equal probably, but there will be these are the 2 choices, big A and little a. And same thing here with the egg. The eggs, there will be a certain probability that it'll be big A, and a certain probability it'll be little a. And so if we do a Punnett square just like we used to do with the individual, but now we're going to apply the Punnett square in terms of a population. It's kind of like a Punnett square on steroids. We're going to use it to understand something much larger than the individual. And so if you look here, when we do this, we know the probability that an individual will have both big A times big A here. That's why we go P squared. It's A times A. So P times P, P squared. Then the probability of having one little a and another little a in this population coming together is A times A, or Q squared. And the reason we go 2 times P times Q is because we have two different ways we can get the heterozygote. Either the egg gives the big A and the sperm gives the little a, or the egg gives the little a and the sperm gives the big A. Same genotype, but two different ways to get it. So that's why we put the 2 there. On the test, any exams you're given will always give you this. We don't expect you to memorize it. So why do we always have to equal 1? Well, we have to equal 1 because the probability here includes all the genotypes in this population. And so it has to equal 100%. It has to equal 1. Because these are going to turn out to be equaling less than 1, but together it'll equal 1. Because it has to include the entire population. So let's work a problem. And I think you'll see they're not too bad. And let's just go ahead and work the one that's in the note-taking guide. And let's write our Hardy Weinberg equation up here just as a reference. And in this problem, we're told, now any problem you may have to calculate the allele frequencies before you can apply the Punnett square. But for now, we're going to just give you this information. We know that this population has 60% CR alleles and 40% CW alleles. And so let's put this in a Punnett square, because who doesn't love a Punnett square? And let's say this up here is going to be what the sperm contributes, and over here is what the egg contributes. And we know that in this population, 60%, or we can put 0.6, of the sperm will be CR. And we know 0.4% or 40% of the sperm will be CW. Likewise, we know that 60% of the population will have, of the female population will produce an egg that is CR, and 40% or 0.4 will have CW. So now we just have to put these together. And again, I don't know if I really stated this very well, but what we're asking in this problem is, if we know this is the percent of gametes or alleles in the population, can we predict genotypes of next generation? That's what we're asking. I don't think I clearly stated that, so I want to make sure that's clear. If we know the allele frequencies, and we do, can we predict what the genotypes of the next generation are going to be? And we're going to do this Punnett square just as I have shown here. So now what we need to do is multiply these. So the probability of, in this population, where 60% of the population has CR and 40% CW alleles, we multiply these together. 0.6 times 0.6 is going to equal 0.36. Of the population, and the next generation will have the genotype of CR. 0.6 times 0.4, 0.24 will be CR CW. 0.4 times 0.6, 0.24 will be CR CW. And then 0.4 times 0.4, 0.16 will be CW CW. And now to complete the answer, what we do is put these together. We'll say in the next population, the next generation, 36% will be CR, or red, we can give the phenotype 2. 16% will be CW, or white. And remember, we have to add these together, because there's two different ways we can get them. So 0.24 plus 0.24 is 48%. So 48% of the population in the next generation will be CR, CW. Now, if you're curious if you did this problem correctly, it's really easy to test, because we know that it has to equal 1. So 0.36, 0.24, 0.24, 0.16 has to equal 1. Or you can look at the percentages here and it has to equal 100%. It's the same difference. So 36% plus 16% plus 48%. If you add those all together, you'll see that you get 100%. So we did the problem right. So we can predict what the next generation's genotypes will be if we know the allele frequencies in that population in this generation. And there is an important caveat to this, or important distinction we should make here. We can only make this prediction if the population is not evolving. That's pretty important to state. That the only reason this prediction can be made is because at this gene here, genotypes, the CRCW genotype, there's no evolution happening on these genes. Alright, so let's state that again, the conclusion there. Knowing that the CR frequencies were 60% and the CW frequencies were 40%, we made the prediction that 36% of the next generation's progeny would be CR, and that 48% would be pink, or CR, CW, and then 16% would be CW, CW. Now we can only make this prediction on a few assumptions. One is that we know allele frequencies of the current generation, and we did. And the other very important thing we need to know, or that we assume I should say, is that the population at these genes is not evolving. There's no evolutionary force upon these genes. If there is, or if there were an evolutionary force on this, then we would not be able to make this prediction. And in short, that's how we determine if a population is evolving. If we know what the allele frequencies are in a previous generation, and if the genotypes aren't predicted as you would predict using the Hardy Weinberg equation, then you would conclude that that population was evolving. So again, we can make these predictions if we know the allele frequencies and if the population is not evolving. If we do this test here and we look at the next generation and we see that these frequencies don't match what we predicted, then one of our conclusions could be that the population is evolving. If they are as we predicted, then we would conclude that the population is not evolving in regards to this gene. Now, this is an important time to bring up the conditions of the Hardy Weinberg principle, because remember we said this only happens if the population is not evolving. And if it's not evolving, we say that the population is in Hardy Weinberg HW equilibrium. If a population is in Hardy Weinberg equilibrium, we can make those predictions. And to be in Hardy Weinberg equilibrium, these 5 things must be true. There are no mutations. So meaning that, in our last example, CR and CW are not mutating. Random mating is the next condition. And again, that's just looking at that Punnett square saying that a gamete with CR, so a CR gamete, doesn't prefer CR or CW of the opposite mating type. Meaning, if a sperm has CR in its genome, it doesn't care if it mates, if it fertilizes a CR egg or a CW egg. It's random. No natural selection. So for instance, there's not a predator that's been introduced to the environment that prefers eating red flowers. So here we would say no selection against red or white flowers. There's no benefit to being red or white, or pink, I should say pink too, in this population. So no natural selection. No gene flow. The population has its gene pool set and no extra genes are being added to the gene pool, and no genes are being removed from the gene pool. So for instance, you don't have a bird or some other animal bringing over seeds for these flowers that are a different color, or maybe more red or more white, more CR, more CW. So no gene flow. And the last one is that the population is large. So over time, as long as the population is large, you don't have to worry about things like genetic drift, where a portion of that population is removed. It's okay if it's well, I shouldn't say it's okay, but as long as that population is large, then we don't have to worry about it having as much of an effect upon the genotype frequencies. Now, it would be very difficult to imagine a population that is in Hardy Weinberg equilibrium for every single gene pair that they have. The Hardy Weinberg equilibrium applies to a single gene pair. The equation only accounts for P and Q. So you could imagine perhaps the CR and CW alleles meeting those 5 conditions. No mutations, random mating, no natural selection, very large population, and no gene flow. And you can imagine that being true, that it applies and there's no evolution in the population in regards to the CR allele and the CW allele. However, that plant has other phenotypes as well. Let's say there is a gene for plant height, and we'll just call it H for height, but you could have a tall plant or a short plant. Well, let's say the tall plants are preferentially eaten by some kind of herbivore, some kind of plant, an animal or insect that prefers the tall plants. So some kind of animal prefers the tall plants. It could be an insect, it could be a rodent, it could be any kind of animal. And because of that, there's going to be natural selection against the tall ones. And so in regards to the alleles, the gene pairs for height, there is evolution. So we're not having natural selection on red or white. That's staying in equilibrium with no evolution. But the height of the plant, maybe that falls out of Hardy Weinberg equilibrium because of natural selection, not in Hardy Weinberg. And we'll say this one is in Hardy Weinberg equilibrium. So within any given population, some of the allele pairs could be in Hardy Weinberg equilibrium and some would not be in Hardy Weinberg equilibrium. Alright, that's it for this objective. If you have any questions, please let us know and we'll be happy to help you out. We'll see you in class. Bye.